Hey everyone, it's your boy here, Mr. Jennings, and welcome to yet another Big Orange Biology. Today, we're going to talk about some examples of cell communication that you can see sometimes even on you. Alright, so we're going to talk about some notable ones here today, and we're going to talk mainly about four. Now, those in my class, you have a note sheet. I recommend using that to fill this out. And for those who happen to be watching, awesome, cool. We're going to talk about this. Now, we have talked a lot about the cell signaling types in class during lecture. This here is just a nice little supplement. Alright, let's talk about quorum sensing. Now, quorum sensing is the idea of reaching a minimum number of individuals in an area, hence the term quorum, if you don't know what the word. It's a political term usually used whenever you have to take a vote, like when Congress is voting or Parliament. In order to make a vote count, it has to be in quorum, has to reach a minimum threshold, and it depends on the legislative body. Now, quorum sensing is the idea that a certain minimum number of individuals are necessary to be there in order for an event to occur. It is communication among microbes that trigger a group response once particular densities are reached. The more dense you are, more likely you're going to elicit a quorum response. Typically works like this, as you see down here. Low cell density means there's a low transmission of the chemical signal that's being submitted paracrine style. So in a paracrine method, a bunch of chemicals are being secreted out. Well, once the cells reach a high density, the cells are going to start absorbing the nearby chemicals as well, as they are also releasing them. And as they absorb enough of the nearby chemicals, you'll elicit the response of a protein synthesis. Or you could also have a situation where enough chemicals are there, you don't synthesize a protein. But this example, we're looking at making a protein. So here's probably a notable example for quorum sensing. We're going to talk about Vibrio fishery. Now, Vibrio fishery is not squid that you're seeing here, not the cephalopod. It is actually a bacterium that lives inside the organs of many marine mammals, namely this cephalopod right here. When the threshold is met, those bacterium will begin producing a light-producing protein, giving the host bioluminescence. So once they have reach a certain amount of bacteria, this Vibrio fishery, you're going to get a nice glowing response. And this is commonly found in most bioluminescent organisms. Been a while. Now, another example of quorum sensing that you can actually see on you yourself at this moment is biofilms. Now, biofilms are when bacterial ecosystems are going to be established and maintained due to this elaborate network of sensing. So bacteria, again, are unicellular organisms. They do not form tissues. They are not multicellular whatsoever. However, they can live in colonies. And sometimes it is very beneficial, very fit of that species to have a sustained ecosystem where the conditions are pretty consistent. So biofilms are going to help promote that consistency. So bacterial species attaches to wherever the host is, makes a colony, and it's going to make a matrix. And what we're going to have is, once that ecosystem starts being established, another bacterial species may move in. And we're going to get a maturation and make this nice big biofilm. And eventually the biofilm will detach and they'll spread again. But what really is a great example of biofilm in the human is plaque found on your teeth. Whenever you don't brush your teeth properly, even if you don't brush your teeth, your bacteria in your mouth are trying to do this all the time, they're going to form plaque. It's actually a collection of bacteria trying to make basically a suitable environment for them. This is ultimately reliant on signal reception. So the cells have to be able to receive the nearby paracrine secreted signals from other members of its species. And that's going to promote more cell division. It's also going to promote nearby cells to kind of congregate together. So it's going to make a nice little plaque. Now, another example of communication is yeast mating, of all things. So yeast mating, first off, yeast live as a haploid. If we don't recall that, or you're watching this and maybe you've already done some haploid diploid stuff, but if you don't know, haploid means you have half your genetic code, half your chromosomes. Example, human has 46 chromosomes. We are considered diploid in that form. The cubic sperm and egg is haploid, meaning they have 23 chromosomes. Well, yeast are one of the many organisms on this planet that stay in their haploid form for most of their life cycle. there's several mini organisms that do this, but yeast is one of the most notable. So yeast is normally found in haploid form, and they have two different mating types. There's two types of yeast in haploid form. There's A and there's alpha. Neither one's superior. Now, each of them, we're going to make their own signaling molecule that they will secrete, again, paracrine style. They're paracrine methods, and they're going to secrete these alpha and A type signals, while the opposite cell is going to receive the opposite signal. So A cells receive alpha signals, and alpha cells receive A signals. And what's going to happen is they're going to create what's called a mating shmoo. Now, this is a term that is dated, to say the least. I am too young to really know what this is. If it wasn't for Cartoon Network in the 90s, rerun channel mostly, I would not know what a shmoo is. In the bottom right hand corner of your screen right now is a shmoo. A shmoo is a Hanna-Barbera character. Yeah. Either way, the scientists who discovered yeast mating, they went, hey, this looks like the shmoo from that cartoon we watch. We have Sonic Hedgehog and we have shmoos. Biology is full of nerds. It's cool. But either way, so we have shmoos. Well, these are shmoos. They're called shmoos because they are elongating. The cell actually will begin stretching its cytoplasm and its cell membrane towards the direction of mating type, based on where the signal is received. It'll actually grow towards the signal. And once they grow towards a certain point, they will fuse, forming a diploid yeast cell. Once that happens, meiosis will follow up. So, unlike organisms like mammals, where we are diploid all the time and we have meiosis to make our gametes, our haploid gametes, and they'll fuse to make a zygote that makes another offspring that's diploid, we'll have a situation where yeast will be haploid, they'll fuse together to form a diploid cell, and momentarily quickly after, they're going to go through meiosis, not mitosis, but meiosis, to quickly make more haploid cells. So it's a form of reproduction. It's a different kind of reproduction. It's a different kind of life cycle that we're normally used to. Now, in my class, we'll be undergoing discussions on meiosis in the following Unit 5, and I'm sure most of your all's AP classes, if you're not in my class, will be undergoing that discussion as well. And last today, let's talk about apoptosis. Now, we're going to talk about more about apoptosis in lecture because it is a major part of the cell cycle, but let's talk about it here. So apoptosis is programmed cell death. Everybody's familiar with that now. It was the first thing one of my AP teachers made me learn about. I remember that. But apoptosis is programmed cell death. Now, programmed cell death is in that way because of the signaling pathway. Well, here on the sim, basically, the cell's going to receive a signal saying, hey, it's time to apoptosis. And that's under normal conditions, but without that death signal, there's going to be this protein called CED9. Don't get hung up on the individual names. Just know it's the name of that protein. Well, CED9 is active. When it's active, it's going to inhibit CED4 and CED3, and that's cool. Well, once the death signaling molecule happens, the death signaling molecule is going to elicit the response to where it's going to inactivate CED9, probably through a G protein coupled receptor. Well, when it's to inactivate CED9, that does not stop. That means CED4 is not being inhibited anymore. So CED4 will then activate CED3 through phosphorylation through a cascade, and CED3 will differentiate into proteases and nucleases. Proteases break down proteins, and nucleases break down nucleotides. So they're going to begin the process of destruction. Now, apoptosis is reliant on cell signaling. There's going to be a variety of factors that control apoptosis, which we will discuss further in lecture. Apoptosis is often regulated by one particular gene known as p53. P53 does a lot more than just apoptosis. It's part of every little thing. And there's more that does apoptosis than just the p53. It's just one small cog in a overall machinery of cell death. Alright, so that's it for today. It's a quick little video. Hope you've had a good time here. Thank you for watching and listening, and have a wonderful day.